What comes to your mind when you hear the term genetic? You gotta turn your volume on. Turn it off. So, what comes to your mind when you hear the term genetics? All right, DNA. What else? What else? That's it? Just DNA from one student? Pedro, what comes to your mind when you hear the term genetics? Genes, very good. Where are genes found? Huh? What are genes for? What are genes for? Nobody? Why do humans, why do animals, why do plants have genes and genetic material? It does allow us to adapt, okay, through various changes. But what's the sole purpose of having genetic information? What do we do with it? We just learned about this unit like last week or the week before. What do we use our DNA for? Look back in your notes. Anybody? Does anybody remember a process called protein synthesis? What is protein synthesized from? What are the two parts of protein synthesis? This is what happens. I miss one day and y'all get mind wiped. That's not what happened. No, don't shake y'all's head yes to that. What is protein synthesis? That's DNA replication. What is protein synthesis? Gerardo. The production of protein from, huh? Thank you, from transcription and translation. Johan, what's transcription?
Whenever like the DNA is making the message through the RNA.
Perfect. All right. So where does transcription occur, Dylan? In the nucleus. Somebody tell me, why does transcription occur in the nucleus? Right, DNA, that's where it is. It can't leave, so it makes mRNA. mRNA goes out of the nucleus into what? Yeah, into the cytoplasm, which has the ribosomes, and it's converted into protein. And that's called translation. Bless you. So our whole reason for having DNA is in order to build molecules, to synthesize biomolecules, and that's how animals get their features. It's also a unit of heredity, which is something that is passed down from parent to offspring. So I know I told you to write the term genetics, but I can't talk about genetics without talking about Gregor Mendel. He's very important to genetics. Do y'all remember Charles Darwin? He was extremely important to what topic? Evolution. He was the father of natural selection. He was very important to that topic. I remember when I was in college, I had to turn a super big project, and it was from my genetics class. I made a C in that class, by the way. But I thought it would be funny if I turned genetics from GE and put JA. Genetics is my first name, JA. My professor took 10 points off of my report. But it was not cool. All right, here's Gregor Mendel. So I want you to write that Gregor Mendel was responsible for, you can just put the study of the inheritance of traits. So write that down. He is the father of genetics. If you want to just write father of genetics, that works as well. He was very important to the topic in the study of genetics. He was the LeBron James of genetics? No? Messi of genetics? No? The Kobe of genetics? MJ of genetics? Y'all get what I'm saying, right? He was very cold at genetics. 224 Hutchins. No, she's not. Okay, I will. All right, moving on. Just write Gregor Mendel, father of genetics. Like he really didn't play any games with those plants. Like he was He set all the standards. Like when it came to plants, he was him with a capital H. Y'all not catching what I'm pitching? All right. So I'll tell you a little bit about Gregor Mendel while y'all are writing that. He was a monk. He was a monastery. So that guy, like, this is where these people, they basically cut themselves off from the world. Everything he did was in the church. So he was actually able to focus on something. All right, here he is. I mean, pretty fly haircut. He was an Austrian monk. He studied the inheritance of pea plants, and he developed the laws of inheritance. His work was not recognized until the 20th century, though, but it was very important stuff. All right. This is an important part. He found that the plant's offspring retain the traits of the parents. I already said he was the father of genetics. We're going to skip past some of this. Now, he was the only person, well, I'm not going to say the only person, but he was one of the first people to document the presence of some biological factor responsible for traits being passed from parents to offspring. Now, here we go. These are those words that you have to know. Write all three of them down. A trait is any characteristics. Heredity, the passing of traits from parents to offspring. Genetics is the study of passing traits. So write these words down in your journal. Good morning. Good morning. Well, y'all look shocked to see me. Y'all was hoping I wasn't here today, huh? You was hoping I wasn't here today? No? Okay. All right, I'll give you about another 30 seconds. So, traits, heredity, genetics. Very important stuff. So hopefully you're starting to see some of that stuff that was on the practice star that you didn't recognize. And hopefully today should be like one of the last days that we introduce stuff that you haven't seen before. One thing I'm going to say that's really important is like the day after the practice star or someday last week or the week before, I assigned each and every one of you a video on mutations. You need to go back and watch that video. That's a small topic, but if you know that, you'll get maybe 2 or 3 questions extra right on your STAR test. All right, my people that are just starting to write, I'm recording this right now, so you'll be able to just go back and watch it. All right, now we're talking about these crosses. Now, this is going to be something that you're going to have to do. We're going to have to learn about how to cross genetics. So there are 2 types, monohybrid cross and a dihybrid cross. So write a monohybrid cross is a cross involving a single trait. A dihybrid cross is a cross involving 2 traits. And go ahead and write the examples. So one trait, that'll be like you're crossing a red flower and a white flower. A dihybrid cross would be that you're crossing a red tall flower with a white short flower. So monohybrid and dihybrid. Raise your hand if this is your first time hearing those words. Monohybrid and dihybrid. Really? Oh wow. Okay. Cool. Y'all didn't work on Punnett squares in the 7th grade? We didn't learn nothing in the 7th grade. That's what everybody said. I already know. This is what you about to say, right? What you about to say?
I've never heard of that.
Oh, okay. Okay. Y'all supposed to. Monohybrid and dihybrid. The prefix mono, what does it mean? One. When else have we talked about that prefix mono? Monomer. Oh, and monosaccharide. Very good. One sugar. What's a monomer? It's one building block. Very good. So right now, dihybrid. What does the prefix di mean? What does the prefix di mean? Two. Dihybrid. Di means two. I don't know if you guys paid attention to that last recording that I did, but I told you guys that there are two ways a cell can exist: as a diploid and a haploid. Does anybody know the difference between diploid and haploid? Y'all got to go back and watch that video. That stuff y'all got to understand. If you don't understand diploid and haploid, I need you to go back and watch that. All right, can I move on?
No.
That's more of a like I'm about to move on. 30 seconds. Okay, moving on, y'all. Remember, you can go back and watch it. All right, here are some more terms you need to know. An allele. Okay, we just talked about a gene. A form of an allele. And before you write, let me explain this. How many, well, of all the traits that a human has, we have 2 genes for each trait. Can some really, really smart student explain to me how you have 2 genes for each trait? Thank you. It's because you have 2 parents. Your mom is going to provide 1 gene. Your dad is going to provide 1 gene. Those 2 genes come together. They fight for dominance, and then you become your own unique organism. That's why there will never be another organism with the same traits as you. Even if you had 100 brothers and sisters from the same parent, y'all would all be slightly different. That's because you're going to get 2 genes for each trait, and those 2 traits, they kind of like shake dice, and that's how people become who they are. It's kind of a random process. But an allele is 2 forms of a gene. Sometimes it's broken down into dominant and recessive. But in humans, it's not that simple. But anyways, so go ahead and write alleles, 2 forms of a gene. I hope you understand that an allele is 2 forms of a gene. It's 2 forms of a gene. You got to understand that. All right. Now, genes can be dominant or they can be recessive. A dominant gene is a stronger gene, and it's represented by a capital letter. So without writing all of this, you can write stronger of the 2 genes, and it's represented by a capital letter. A recessive gene, it's weaker, and it has a lowercase letter. So I wouldn't even write all that. I would just write weaker gene, lowercase letter. Weaker gene, lowercase letter. And that joke, that's supposed to be a joke of the top designer genes, you know. I told you guys, like, see somebody, be like, hey, nice jeans. And I'm not talking about no Levi's. You raise your eyebrows? Yeah. I didn't teach y'all about that. Oh, all right, cool. Trying to teach y'all something. If they don't understand nice jeans and they're at this grade level, at this age level, you don't need to be talking to them because they're not on grade level. Oh, what are you doing? That's not a good one? No? All right, fine. I'm trying to help y'all out. All right, moving on. All right, more terminology. Now, I know this is all new, but this is that stuff that's going to get you those extra points on that. Yes, sir. Yes. But the words that we're about to go over, we're going to go over two words. One of them is genotype, which is an organism's genetic makeup, and the other one is phenotype, which is a physical makeup. All right, so we're about to go over it right now in a second. But I'll give you an example really quick before we get to the definitions. You cannot see an animal's genotype. The genotype is not always visible. The genotype exists in the chromosomes. It's not always expressed. So some people in here can carry the gene for blonde hair, but you don't have that trait. So sometimes in couples you'll see, or even in when we're breeding animals, we'll see like a litter of brown puppies and one white puppy. That's because both of the parents had that recessive gene, that weaker gene, and by chance those two weaker genes got together, and that's the trait that was expressed in the offspring. So genotype, it's the genetic combination. The phenotype is easy, physical feature. So your genotype is always going to look like this. You're going to see either big R, that means they have two dominant traits. Big R, that means that they're a hybrid. Or little r, that means that they're recessive. The phenotype is going to be red, white, pink. The phenotype is going to be a physical thing that you can see with your eyes. That's a phenotype. I think.
Oh, yeah.
Go ahead. Here, take that. I'm recording this. Watch this. Write the notes.
Yes, sir.
So genotype, genetic combination. Why do I have two letters representing a gene? Why do I have two letters representing each a gene? Yeah, because you get two genes, one from each parent. Now, if y'all can remember back to our last lesson, remember everything piles up. So if y'all can remember back when we did this, the meiosis, I told you when organisms make reproductive cells, they divide their chromosomes in half. So here's an example. How many chromosomes does a human have? Now, how many chromosomes total does a human have? 46. Very good. Yeah, we have 46 chromosomes. So in the reproductive cells, how many will be there? Yeah, let's have 46. 23. So two reproductive cells get together and it makes two reproductive cells get together and they make an offspring that has a full set of chromosomes. Okay. All right, we got a couple more terms. All right, so if we're looking right here, it says the genotype of alleles. If big R is a red flower and little r is a yellow flower, you need to know that this is how those possible combinations are going to look. Big R means they're dominant. Big R means you're a hybrid. Little r means that you're recessive. Now, here's more words. I know this is word heavy, but once you get these words, you'll be good. All right, a homozygous genotype. A homozygous. So the prefix homo means same. So homozygous, that means that they have the same gene for each trait. So that prefix, same. So same genotype. So homozygous means that the genotype combinations involve two dominant or two recessive genes. So you can just write that. Sometimes this is called pure. Pure. Now, a heterozygous, the prefix hetero means different. That's a gene combination of one dominant and one recessive, and that means hybrid. So a hybrid. So hetero, different. Write these examples. So big R, little r, that's the only type of heterozygous that we know for monohybrids. It'd be one capital, one lowercase. And make sure you write that word hybrid. Can I move on? Just about? All right, moving on. All right, so now a Punnett square. A Punnett square. A Punnett square. So, right, a Punnett square is used to help solve genetic problems. If you go back and you look at the last recording, that's going to be in the Monday folder of week 8, 39 weeks. You'll see that female gametes and male gametes, those are just reproductive cells given by those parents. We use a Punnett square to put those reproductive cells in there to determine the probability of offspring. Breeders will probably do this to determine how many rare puppy breeds they will get. Like breeders, they love to breed pure puppies. Some of them like to breed mixes. I have a dog at home, and he's what you call a genetic abomination. But he's cute. He's very cute, but he is an abomination. I'll tell you that. Why is he an abomination? Well, his dad was a toy poodle. So his dad was like this big, very angry, very feisty. His mom was a golden retriever. So they take those and they breed, they're called doodles. So they're like a golden retriever mixed with I don't know what's wrong with that dog. I really think that messed him up, but he's cute though. So I'll keep him. All right, so let's just say, boom, I have a Punnett square. Now, let's just say I told you, let's do a real quick practice problem. Let's say we're breeding dogs, okay? Let's say that I had a key. Every time you do a genetic cross or something, you're going to have a key. Or you're going to have to make your own key. So the key means for black fur and then for white fur. Okay? Let's just say that the black fur is dominant. I'm going to represent the black fur with a big B. What letter would I use to represent the recessive white fur? Lowercase b, very good. You don't want to use a W, that'll mess you up. So what we're going to do is say that the mom is homozygous dominant. So if the mom is homozygous dominant, what two genes does she have? Huh? Thank you, 2 uppercase B. So that's mom. You know what? Let me do the mom some justice and write it in pink so we don't get it. So big B, big B. All right? And then we got the dad. What are his 2 genes? If he's heterozygous, what are his genes? He's heterozygous. Huh? Yeah, big B, little b. So now let's cross the mom and the dad. All right, so let's see. Now the reason we use a Punnett square like this to represent probability is the same reason this. How many pennies is it? 100. So it's 100%, right? How many quarters? So this is divided into 4s. How many quarters gives you 100%? 4 quarters. So it's the same way. Each of these quarters. Can you go higher than 100%? 100%. When we're talking about a dollar, can you go higher than 100%? No, 100% is the max. So even if they had 1 million children, they would all have to fall into this probability scheme. Now let's put the parents' genes into the probability scheme. Female gametes, these are the egg cells that the female makes. So since her genotype is this, she's only going to make pure eggs. So her reproductive cells are only going to be big B, big B. That's it. She can't make any recessive genes because she's a purebred animal. Now the dad, he's not purebred. He's a hybrid. He's a mix. or some people call it a mutt. Now you take these genes, the way you do this, you bring this one down, this one over. So let's do it. I'll take the mom's genes, big B. I'll bring the dad over, big B. I'll bring the mom's genes down here, big B. The dad's genes over here, little b. And you keep going like that until you get your Punnett square solved. And then little b. That's it. So this is the probability of their puppy. So let me tell you. So if they had, well, let's see, what percent of their puppies will be purebred? Thank you. 50%. 50% of their puppies will be homozygous dominant purebred. How many would be heterozygous? 50. How many would be homozygous recessive? 0, because they don't have any that are little b, right? So this is how you do a Punnett square. This is how you're going to have to be able to add math into these genetics problems. So I would say, let's just say they had a litter of, let's say they had 8 puppies. If they had 8 puppies, what would the genotype be for the 8 puppies? So you would say the genotypes are big B, and big B, little b. So of the 8 puppies, you would say 50%, and then the other 50%. Those numbers add up to 100%. So 4 would be big B, and 4 would be big B, little b. That's how that works. All right. Let me just check your worksheet and see if there are any terms that I have to go over. Punnett square, monohybrid, the law of independent assortment, dihybrid. So I do want to go over this word, the law of segregation and independent assortment. We'll skip to that, and then I'll wrap up the recording. We already talked about plant reproduction. Y'all remember all those terms, all those jokes I used? Okay, very good. I don't have to say any of those jokes again? No? Yes, maybe? Y'all don't want me to do it. Now, monohybrid crosses are easy. You're going to get some practice with it on the worksheet that y'all do Friday. But today is more just to get you introduced into the genetics. The vocabulary of genetics. Sometimes you'll see this right here. Whenever you see P1 generation, that's the parental generation. I'm going to stop the screen right here just so y'all can pause it and read through it. If you see F1, that's the first generation. F2, second generation. But read over it. All right, so here we go. Here's another, the trait that we're breeding, so this will be our key, is seed shape. The alleles, what's an allele? What's an allele? 2 forms of a gene. So we have big R, which is round, little r, which is wrinkled. Which one is dominant? Which one is recessive? Wrinkled. All right. So let's say we're breeding a homozygous dominant and a homozygous recessive. This one doesn't say which one is which parent, but still, we just breed these two together. We get 100% what? What would you call that? 100%. Huh? Heterozygous or hybrid. Very good. Okay, let's skip to the here we go. So these terms. So we have 3 laws: the law of dominance, the law of segregation, and the law of independent assortment. Let's get these terms and this should be wrapped up. All right, the law of dominance. This just means that you don't even really have to write this down. This should just be brainless. The gene that is stronger is going to always dominate the recessive gene in simple crosses. We have not talked about the other types of crosses, which we're going to talk about later. But as far as what you need to know right now, big letter equals stronger trait. That means that that gene is going to be what is seen whenever it's mixed with a recessive gene. So you don't have to write this, but just look at this. Big R, little r, 100% are going to be that heterozygous, but for their phenotype, what is a phenotype? What is phenotype? Huh? Their physical traits are going to be all dominant. They're all going to have that, but they carry the recessive gene. So let me show you what would happen if I cross two heterozygotes. Matter of fact, y'all can practice. So on this side right here, on your paper, I want you to cross two heterozygotes. Cross two heterozygotes. Cross. Come on, let's do it. Cross two heterozygotes. Make the square thing, the Punnett square. First thing you're going to do is make a key. Big R. What was big R? Big R was round, right? What was little r? Wrinkle. All right. So let's say we're crossing two heterozygotes. So our second step would be to find the mom and dad genotypes. So if they're both heterozygotes, what are their genotypes? Yeah. Well, I only got Dante and Dylan participating. Why is that? Help them out. Naomi, you're helping out too. Sorry. So your peers are gonna hear y'all's names and be like, "Oh snap! Oh my boy's smart." You know how they do? Or they just gonna hate. They're gonna hate. They're probably just gonna hate on y'all, to be honest. It's okay though. Big R, little r. Big R, little r. Now I want y'all to do this cross. 624 Hutchins. Good morning. Fifth period. What do you mean? No. Okay. Yeah. All right. All right. Anyways, big R, big R, big R, little r. You bring this one down. Always put your capital letters first, and then boom. So now this one, if I told you to tell me your genotypic ratios, this is how you would find it. So your genotypic ratios would go like this. So your genotypic ratios, we go like this. So homozygous dominant. I'm just gonna write 8. Nah, I gotta write it out. Homozygous recessive. And then heterozygous. Okay. So what your ratios would be, and I'm going to go ahead and write out what the genes are. So that would be big R, little r, and then big R, little r. How many would be homozygous dominant? 1 out of 4, which is equal to what? 25%. Very good. That's 1/4 out of $1. How many would be homozygous recessive? 1 out of 4. That equals 25%. And how many would be heterozygous? 2 out of 4. What's the percent on that? 50%. Y'all are smart. Y'all got it. All right. So that's how you would do these problems. I know it's not the easiest thing now, but practice makes perfect. Okay. So if you need to write anything down from that, go ahead. I gotta check that email more.
You do?
This signature right here.
Uh-huh. That's
Is
it.
this okay?
Yeah, nothing has changed.
Okay. One of them days for me.
It is. How did you get ready this morning? Yeah, that's right.
Do we need the date?
No, should date it.
Okay.
Yes, sir. But yeah, that's why I brought it up. You can see it.
I appreciate you.
You're welcome.
All right, everybody got that? All right, so moving on to our last thing. Oh, no, we had two more actually. All right, the law of segregation. Now, this one I want you to write. The law of segregation means that basically during the formation of gametes, remember gametes are the egg and sperm cells, the two alleles responsible for the trait separate. So just write that top part. So during the formation of gametes, the two alleles separate from each other. Okay. Now moving on to the last thing. Oh. All right, everybody got it? All right, the law of independent assortment. This means that alleles for different traits are inherited or distributed independently of each other. So alleles for different traits. So if we're talking about height and flower color, those genes are not, they have to be separated before they can be theoretically inherited. So alleles for different traits are distributed independently of one another. So whenever we're talking about independent assortment, that's when we're moving on to dihybrid crosses. And there's a whole process that we have to use in order to get the gametes for dihybrid crosses, but we're just going to leave it at that for now, and we'll get there eventually. All right. So this is the last slide that you really need. This is recorded. I'm going to upload it. But this slide, along with what I uploaded into Schoology, should help you get through this packet right here. Any questions? No questions? Nope. Y'all having a good time out there? Science is fun. Biology is good. Biology is lit, as the kids say. It's not lit. It's not. All right, fine. I'll leave y'all alone. All right. Nobody want to say nothing to y'all's peers before I stop the recording? Okay.